much to Dr. Lebois and Steve Ramirez for coming. What we're going to do is Evan Lebois is going to talk first, and then we will hold off questions. Then we'll go to Steve's talk right away, and then after we'll be able to ask questions to both speakers. So I'm going to do just a quick intro on Dr. Lebois, and then I'll do the intro before Steve Ramirez comes up. And you guys are going to fill in anything I forget as I brag about you too. Okay, so Dr. Evan Lebois, he started out in the basically in the drug discovery program at Vanderbilt University, where he got his master's. There he was studying neuropharmacology, specifically Alzheimer's disease, but also other CNS disorders. He, importantly, this is probably the most important thing they said I need to mention, is he runs hundreds of miles normally, in races that are hundreds of miles, so that is important. So he brought lots of the patents that he developed at Vanderbilt when he then transferred to Emory University, where he completed his PhD. In neuroscience, he worked with Dr. Joe Manns, doing in vivo electrophysiology, and also Dr. Alan Levy, who heads the Alzheimer's Disease Center, a really big Alzheimer's Disease Center at Emory University. And so he was doing behavioral pharmacology with him as well, continuing his drug discovery research. And he is published in dozens and dozens of journals, especially for being such a young investigator. So right now he is actually heading his own lab at Pfizer. So he actually heads his own neuroscience laboratory where he has a couple people working for him, working on several psychiatric disorders. Primarily, he's still continuing Alzheimer's disease research, but also expanding more on schizophrenia, depression versus PTSD, and really sees sort of all aspects of the scientific process. So from studies with rats early on, and then helping to design experiments for primates and even humans. So he's really there in all stages of drug discovery and really translational neuroscience. So especially to my students who will get time to chat with him afterwards, please use him as a resource for talking about work in industry and neuroscience.
I was also a chemist in my former life, so literally any questions is fair game.
He knows a lot, so he's going to be a great resource. So we welcome Evan, Dr. Lebois, thank you so much for coming. And thank you all.
Thank you guys so much for having me. This talk is really going to focus on work pre-Pfizer, so I'm not here on behalf of Pfizer in any capacity. This is all work I did as a master's and PhD student that I'll talk to you about. It focuses on basic mechanisms of memory, but also how those intersect with Alzheimer's disease and what happens when things go wrong with aging and disease, and how memory processes break down, and really what we can do therapeutically to help restore memory function. So I titled my talk, When Memory Betrays You, How Memory Declines with Aging and Alzheimer's, and what we can do about it. Before I really launch into things, I want to go over this brain structure that's all near and dear to our heart, the hippocampus, and where it lies in the processing hierarchy of the brain. And specifically, the fact that what ends up happening is non-spatial and spatial inputs come in through the entorhinal cortex to the hippocampus. And an important part of this diagram is that this information stays really segregated, that is, non-spatial information stays separate from spatial information until the level of the hippocampus. And it's only in the hippocampus that this information gets combined together to form episodes. And the essence of episodic memory is really what happened where. And interestingly, it's now being appreciated that hippocampal cells are also modulated by time as well. So that really is what happened where and when. And today, Steve and I will really focus on the what and where of this. And next week, you have Nick Robinson coming in from Howard Eichenbaum's lab, who's going to talk a lot about the temporal modulation of hippocampal cells. So before I launch into this, I have to give my generic conflict of interest statement in the fact I work for Pfizer now. They're a for-profit entity, but none of this work focuses on what I do at Pfizer. It was all conducted at Emory and Vanderbilt. And also, I'll talk to you today about some cholinergic drugs that I developed while I was at Vanderbilt, and I have intellectual property on those drugs. So that said, my former lives, as Vicky mentioned, I did my master's at Vanderbilt, where I was very involved in drug discovery. And this compound here is actually the subject of what I'll talk to you today about, therapeutically speaking. It's a cholinergic agonist for M1 muscarinic receptors. And after Vanderbilt, I took these drugs with me and decided to branch out into various biological testing from there, and worked with Joe Manns to do systems electrophysiology experiments to ask how cholinergic activation shapes memory circuits and what the hippocampus does. And also worked with Alan Levy in the Alzheimer's Center to ask, do these drugs help prevent Alzheimer's-like pathology and memory impairments from forming in animal models of Alzheimer's disease? Alzheimer's disease in general is a huge, huge problem, and it's likely to become one of the biggest medical epidemics that we all face in our lifetimes. Approximately 1 in 8 people over 65 have full-blown Alzheimer's disease, and if you make it long enough to 85, that goes up to about 50%. Currently, Alzheimer's represents about a $180 billion healthcare burden, and part of the tragic part of this disorder is that it's very chronic and protracted. People often live about 10 years or more. So from stress on the caregiver standpoint to financial burden on the healthcare system, this is an extremely burdensome disease. Something to realize is that only about 1, maybe 2% of Alzheimer's disease is actually hereditary or has a known genetic basis. Approximately 99% of the disorder is completely sporadic. And that's important because all of the animal models that we have are based upon these 1% known genetic mutations. No treatment is currently available to halt disease progression, and recent research, we're now appreciating that this disease likely, the cascades that lead to eventually Alzheimer's begin as early as your 30s or 40s. So if we want to prevent this disease, we really have to understand these early processes and be effective early. So pathologically speaking, what is Alzheimer's disease? It's a progressive neurodegenerative disease, so the brain physically dies off with Alzheimer's disease. Shown here on the left is just an example of a healthy brain hemisphere contrasted on the right with a brain hemisphere that from a severe AD patient. And you can see there's marked degeneration here, enlargement of brain ventricles, hippocampal atrophy, and widening of your cell side. Early on in the disease, or just with the disease in general, cholinergic neurons are preferentially susceptible. So acetylcholine is a very important neuromodulator in the brain, responsible for modulating memory circuits. And for whatever reason, a lot of these neurons are lost with Alzheimer's disease. So one of the prevailing hypotheses for treatment has been to restore acetylcholine function, and that's the focus of the drugs that I'll talk to you about in a little bit. As I mentioned, life expectancy is usually around 10 years, and the earliest observable symptoms people come into the clinic with are memory impairments. So often, people will be driving around and just suddenly not remember where they are, or they'll get lost somewhere very familiar, and this triggers alarm bells in their head, and they go in and see their neurologist, and only then they're discovered they have Alzheimer's pathology. Molecularly, Alzheimer's is really identified by 2 major hallmarks. There's these beta amyloid plaques that you hear a lot about, as well as neurofibrillary tangles. And these plaques are composed of amyloid beta peptides that stick together and form these extracellular plaque aggregates. The tangles that occur with the disease are intracellular accumulations of this protein tau. It's normally a microtubule binding protein that helps stabilize cytoskeletons. And I'm not going to talk at all about tau today, but I'll mention that these plaques that form, they're actually not correlated with cognitive status in Alzheimer's disease at all, but what is very correlated with the cognitive status is the level of these soluble amyloid species. So these are really the precursor molecules to the plaques. But since these are soluble, they can diffuse throughout the brain and disrupt synaptic plasticity. And I'll mention that more as the talk goes on. But it's thought that, especially early on in the disease, these are the major molecules at play. As I mentioned, there's a declining cholinergic function, but not only with Alzheimer's disease, just with aging as well. So really, there's a dysfunction that begins with aging, and then cholinergic neurons eventually die off with the disease. But the idea is that if you can restore this lost function, you can recover the memory that's damaged due to Alzheimer's disease. So acetylcholine, I mentioned, is an important modulator of memory circuitry, and acetylcholine acts on a couple different classes of receptors. The major ones I'll focus on today are these muscarinic receptors, and these mediate the slow modulatory actions of acetylcholine. They're G-protein coupled receptors. There's 5 major subtypes, M1 through M5. They're very similar to one another. That is, they all are engineered to bind acetylcholine. They're localized throughout the CNS and the periphery, but within the brain, M1 and especially M4 are the major subtypes of receptors, and they're crucial for mediating learning and memory processes. Importantly, for us, M1 is heavily expressed in the hippocampus, but also present in the basal ganglia and neocortex. And we know that taking away M1 impairs memory in animals, and activating M1, either genetically or pharmacologically, leads to memory improvement. So it's, therapeutically speaking, an attractive target to try and modulate. In addition to its beneficial effects on memory, M1 activation also exerts beneficial effects on Alzheimer's disease pathology. That is, M1 receptor activation can lead to activation of intracellular signaling cascades that impinge upon the proteolytic enzymes that actually process this amyloid precursor protein. And it's the cleavage of this precursor protein that gives rise to the amyloid beta peptides that you see in Alzheimer's disease. I should mention that these secretase enzymes that process amyloid precursor protein are also big direct targets of interest as well to try and block the accumulation of amyloid. But amyloid precursor protein, or APP, can essentially be processed in one of 2 ways. Either this amyloid plaque forming direction or this other direction where soluble extracellular fragments are produced that can just diffuse off and don't aggregate. And the effects of M1 activation lead to a biasing away from the production of amyloid and promoting the formation of these soluble molecules. So you actually blunt the ability of these molecules to aggregate together and form A-beta peptides. So this indicates that M1 activation has disease-modifying potential and could actually modify the pathology of Alzheimer's disease. So how do we measure memory? One way that we do it in animals is to actually use Morris water maze, for instance. And this is a very sensitive test of hippocampal memory. Hippocampal lesion rats are very impaired in this task. And what it involves is essentially a pool of water with an escape platform submerged just below the surface of the water, and various contextual cues placed on the wall or even within the testing room, demarcating the different quadrants of the pool. And this red X indicates the point at which you might place the animal into the pool. And initially, when you first place the animal in there, they swim all around and eventually by sheer luck find the platform under the water. And rodents really hate being thrown into a pool of water, so they very quickly associate in a few trials where that platform is relative to the spatial cues. And animals that have hippocampal damage always look like this, regardless of how many times you throw them in the pool, so they can't acquire this task. Another way we can measure memory is actually by in vivo electrophysiology. And this is something I'll talk to you a little bit about as well. This involves building these microelectrode arrays where each of these screws on this array can be independently turned and adjusted, and these microelectrodes projected down into the brain. And shown below here is a slice through the dorsal hippocampus with CA1 and CA3 fields demarcated. And I actually did a project where we record from CA1 and CA3 simultaneously while animals are doing various tasks. So we can actually visualize the brain activity in real time by doing this. So the two things I really hope to tell you today are just overall to convey that M1 agonists can bolster the function of memory circuits, but also act to protect against cognitive decline that's relevant to disease, in particular Alzheimer's disease. And first, I'd like to demonstrate that M1 activation actually biases the hippocampus away from coding for familiar information towards coding new and current information. And these are experiments that I did in healthy adult rats. And then second, I'd like to tell you about some work I did in transgenic Alzheimer's mice to demonstrate that chronically activating M1 actually prevents Alzheimer's-like pathology from forming and prevents memory impairments from occurring in these mice. So up first, does M1 activation bias hippocampal circuits towards the coding of novel information relative to familiar information? And in order to understand this, I'd like to talk about this important process that the hippocampus does called pattern separation. And broadly defined, pattern separation is the ability to differentiate something new from something similar. So that is, I've given some example stimuli here, and probably everyone in this room can tell that the top row is different from the bottom row, but that they're pretty similar. The important thing is here that people who have age-related cognitive decline, Alzheimer's patients, they have a lot of trouble disambiguating this bottom row from this top row. And that's an impairment in pattern separation that happens with aging. And the expectation here is that a drug that's good for memory should help you discriminate closely related stimuli from something you've seen before. So you should be able to tell which is different. And I'll just mention that several different diseases and different disease states have deficits in pattern separation associated with them. Pattern separation breaks down in mild cognitive impairment, early Alzheimer's disease, age-related cognitive decline, schizophrenia. People with PTSD and depressed patients also have deficits in pattern separation. So it has broad utility in being an index of memory function and hippocampal fidelity, and serves as a great marker therapeutically. If you can improve pattern separation, you should be able to improve memory performance. So in order to measure pattern separation, we've used an in vivo electrophysiology approach to doing this. And you can essentially record two different types of data with in vivo electrophysiology. We get action potential data, so these are spikes that we record extracellularly from neurons, and we also simultaneously record the local field potentials, or LFP oscillations, from neurons. And I'm not going to talk at all about LFP today just because of time constraints, but I will talk a little bit about the action potentials that we record. So hippocampal pyramidal cells are often referred to as place cells, and the seminal observation that led to this, as their name would imply, is that they form place-specific firing fields. So this is an example of a rat running around just a square box for 15 minutes. And shown in red here is every time this particular cell elicits an action potential during that 15-minute period. And in black, in the background, is where that rat actually physically occupied in the enclosure during that 15 minutes. And what we can do is actually transform this and plot it as a rate map to depict the firing rate of the cell over time. And we can see that the cell really prefers the bottom right-hand corner of this enclosure. And so this is one hippocampal pyramidal cell, and I should mention that due to the size of the arrays that we use and the number of microelectrodes, we can record from, say, 100 neurons simultaneously. So I could generate 100 different plots like this and ask what they look like and what the drug does to them. The really neat property of place cells that we want to take advantage of is that not only do they form place-specific representations, they change in response to contextual inputs. So that is to say that I have a particular representation of this room, and if I were to go next door, the room might look similar, but composed of different items. So I'll form a new representation in the different room, but due to the fact some things will probably overlap, some cells will stay the same. But largely, the cells that are active in this room will change either their firing rate or location that they fire in when I go to the upper room. And if we actually take a rat and run him through a sequence of shapes for 15 minutes each, starting in a square, going to an octagon, a hexagon, a circle, and then reintroduce him to a square, we can look at the place cells and actually see that they change. And what's been demonstrated again and again across different labs is that you form an initial representation, this representation changes in a graded manner, and then you recover the initial representation once the animal's in the context that you started in. So the fact that the representation looks the same in the square means the animal knows where he is and recalls the same map in order to represent that environment. What we'd like to see is that when giving a drug that's good for memory, so like an M1 muscarinic agonist, do rats actually have an enhanced ability to represent these novel enclosures relative to somewhere familiar? And what we can do is actually correlate the activity in these middle enclosures relative to the initial enclosure and express that in a ratio of activity in the middle enclosures relative to the initial and final enclosure. So what you get with this spatial discrimination is really the degree of change over those initial and final enclosures. So the higher the number, the more the animal is distinguishing those middle enclosures. And what we actually see is that with increasing doses, there appears to be a sweet spot in this middle dose of enhancing the animal's ability to code for those middle enclosures. So we're actually biasing the hippocampus away from encoding the familiar enclosure and pushing it towards increased coding of new information. Now, I'll note that this green column over here is actually a drug donepezil, which is an acetylcholinesterase inhibitor. And it's just a non-selective way to increase acetylcholine in the brain. And this is actually the main standard of care for Alzheimer's disease. And I'll also mention it's not very effective. So in terms of the standard of care currently for Alzheimer's disease, we're doing a lot better in terms of urging the hippocampus to code new information. And like I said, we're able to record from a lot of cells. So this is actually aggregate data of 150 different hippocampal pyramidal cells. So it's not like we just look at a couple of cells and plot these effects. To summarize, if you activate M1 receptors, you can actually bias hippocampal circuitry. And there's a whole lot of work here I didn't talk about with the local field potentials and understanding how we're biasing hippocampal circuits, but I'd be happy to talk to you about all that work later if you'd like. We're able to increase the spatial discrimination of pyramidal cells and help them track contextual change better by driving the cholinergic system. And this really suggests that M1 agonists can actually offer a beneficial treatment avenue for disorders that have pattern separation as a hallmark component. The second aspect of what I want to talk to you about is just driving the same cholinergic system that biases hippocampal circuits to code new information. Does that actually prevent disease pathology in a transgenic mouse model of Alzheimer's disease? And the mouse model that we picked is a particularly aggressive mouse model, termed the 5X FAD mouse. And just historically, how these mice are created is, I mentioned, they take human genetic mutations and put them in a mouse, and the mouse pumps out amyloid protein and accumulates amyloid plaques, like you would see in an Alzheimer's patient. What they did in this 5X mouse is they took 5 familial mutations, so if something works, why not put more mutations in there? And this mouse really develops a brain full of amyloid, like a huge, huge amount. And it gives us a nice, robust window in which to detect drug effects then on amyloid. So these mice start accumulating amyloid pathology around 2 months of age. And by 6 months, we know that they're memory impaired. They develop marked impairments in the Morris-Water maze right around 6 months. So what I wanted to do is to really ask the question, does chronic treatment prevent? And what we did was actually go into the mice before they develop any detectable pathology, start administering drug, and then test them at a point we know they should be memory impaired, and ask, are they memory impaired, and what does their brain look like? And a crucial aspect of this is that we allow drug to wash out for 24 hours from the system of the mice. And this is important because any effects that we detect on memory or disease pathology, we want to be due to having modified the underlying pathology, not symptomatic improvement. So if the drug is still on board, it could be just symptomatically benefiting the mice, and that'd be hard to tease apart from any true underlying pathological effects that you have. So it's a long experiment. The total time course here is about 6 months, and the results we obtained, though, were incredibly exciting. So when we tested the memory of the mice in the Morris-Water maze at 6 months of age, shown here in the gray are mice that were dosed just with regular drinking water this entire time. Shown in pink are the mice that were transgenic, but dosed with drinking water. So relative to just healthy wild-type mice, these mice were significantly impaired in the Morris-Water maze. And in blue were the drug-dosed mice relative to the memory-impaired transgenics as well as the wild-type. And they were indistinguishable from the wild-type. So no significant difference there, and significantly improved relative to just the transgenic mice. And importantly, no deficits were observed in the motor impairment, or the motor activity of these mice. So they looked and performed and swam and locomoted just like wild-type mice. So our drugs weren't having any side effects there. So incredibly exciting. And what does the underlying brain pathology look like? Does this actually correspond with improved pathology? And in order to do that, I essentially took all the brains and processed them in one of two ways. Serial section through them so that we could stain for amyloid pathology. And here we're just visualizing the plaques that form in the brains of these mice. And the parallel processing pathway was just to homogenize these brains, grind them up and mix them up. And that enables us to stain for the soluble amyloid species that you can't detect really well with antibodies in sections. And when we actually sectioned through the brains of these animals, just to cut to the chase, what we saw was a significant improvement in terms of reducing the pathology present. So if you look at the top two rows here, these are representative examples of high expressors, medium expressors, and low expressors from just vehicle-treated transgenic animals. And these are all cortical sections in the top row, so this is through frontal cortex. And you see a high level of amyloid accumulation that tapers off in the low-expressing animals. So there's a pretty good range of amyloid expression here, but the maximum expression is pretty high. And same goes, these are all hippocampal sections through just vehicle-treated transgenic animals. And in the bottom two panels are the same thing for the cortex and the hippocampus of drug-treated animals. So relative to the vehicle animals, these drug-treated animals show significantly less amyloid pathology. You'll note that it's still present, so we haven't totally eliminated the amyloid pathology, it's just significantly less. So if you actually quantitate that for the cortex and wild-type animals, the transgenic animals relative to them have significantly decreased levels of amyloid once dosed with drug in both the cortex and the hippocampus. So in the hippocampus, we're looking at like a 20% reduction, and the cortex closer to a 30% reduction. And if you actually grind the brains up and look at the soluble amyloid species, I mentioned before that it's these soluble amyloid species that really correlate with the cognitive status of disease and memory performance. So if we stain, or if we look at these soluble species in ELISA, for instance, we can actually detect the level of soluble amyloid beta present. And again, in the cortex, in the blue, drug treatment significantly decreases amyloid, or soluble amyloid in the cortex as well as the hippocampus. And if you correlate these results with the behavioral performance on the Morris-Water maze, for vehicle-treated animals in the pink, there's a very robust correlation between high levels of amyloid and low levels of behavioral performance. And this correlation is completely abolished by M1 agonist administration. So reducing the amyloid is able to break this correlation. To summarize, M1 activation has disease-modifying potential for treating Alzheimer's patients. It's an incredibly active area of research that companies are very interested in for treating Alzheimer's patients. In a preventative manner, which is very exciting, this is really the first evidence of a chronic administration preventing and delaying memory impairments from forming. So at 6 months of age, you can prevent memory impairments in these mice and significantly blunt amyloid pathology by decreasing both soluble and insoluble amyloid species. I didn't talk a lot about the insoluble experiments, but the same holds true for insoluble as well. But mainly this provides a critical proof of mechanism that chronically activating M1 is a viable preventative strategy to pursue for Alzheimer's disease. And I'll mention that the higher soluble amyloid levels, particularly in the hippocampus, we know significantly impair synaptic plasticity, which is why they correlate with worse memory performance. So I'd like to acknowledge several people, my former PIs at Emory, Allen and Joe Manns. The folks I worked with at Vanderbilt were instrumental in this work. They actually provided all the drugs for us to use at Emory. Jeff Kahn and Craig Lindsley, and of course, all the funding that made this work happen. So I'd be happy to take any of your questions. This was one of our volunteers for the electrophysiology experiment. We put them on these stools and trained them to eat Fruit Loops. They're happy to comply. So, thank you.
Time to do just a couple questions now, and then we'll go into Steve's talk, and then we can ask questions to everybody at the end. So maybe a question or two.
In terms of clinical trials, have you started that?
So we are now interested in heading in that direction. We haven't started clinical trials yet. That's definitely something we'll be looking to do in the next few years.
Part of your patient population, obviously, your studies for representative, would you do that risk population or somehow see the amount of black?
So yeah, so that's a great question.
Repeat the question.
So the question was that when moving towards human trials and thinking about designing clinical studies, how do we pick a patient population? And this is true not only for Alzheimer's disease, but for any psychiatric indication. They're very heterogeneous. They have no known genetic cause. So how do you identify an at-risk population? And one of the best things that's actually been done, so we can image for brain amyloid in living people. There's actually tracers out there that bind to amyloid in the brain and enable you to see it. So that can be used for inclusion criteria. But as for cognitive status, there's actually really nice technology that's been developed that tracks eye movements, and you can do a visual pair comparison task where you show someone an image, you take it away, and then you show them either the same image or a different image. And if it's a different image, there will be increased fixation on the image details relative to the old image that they just saw. But if it's the same image, people really don't care as much, and they tend not to look at all the details. And you can actually use that with enough images to stratify healthy people from mild cognitively impaired, so this is the precursor to Alzheimer's disease, from Alzheimer's patients. And even within the mild cognitive impairment group, that's a group where it's basically a catch-all clinical diagnosis. So some of those people are going to develop Alzheimer's disease. Some of them are going to be just fine and just have age-related memory issues. So you can use this eye tracking task to actually pick out in that MCI basket who's going to convert to AD within 3 years. So we would probably utilize some approach like that in order to pick out a group that's at risk and then put them on treatment. Because at that point, they have significant memory impairments, but not a lot of degeneration in their brain. So there's still a lot to take advantage of there with the drug, but the neuron that hasn't happened yet. All right, thanks a lot.
So while Steve sets up his talk, I'm just going to give a brief introduction for Steve Ramirez. Thank you again for coming. And again, we'll get to do questions for both of the speakers after. So Steve Ramirez is currently at MIT. He actually doesn't have his PhD yet, but he's so accomplished already. 2 more months. So he already has dozens of publications, even though he doesn't have his PhD yet. He's working right now in Susumu Tonegawa's lab, pursuing his PhD. He started, just like you all are starting as undergraduates, that's where his passion started, in Howard Eichenbaum's lab at BU, which is another really spectacular lab in the neurobiology of memory, and we have somebody from his lab coming next week to talk to us, actually. So he started off there. Now he's at MIT. Recently, he's been given many awards, so I'll mention just a few. He's now the Harvard Society of Fellows. So he'll be starting up his own lab, applying for grants probably in the New England area in the next few years. So we're excited to see where he ends up and who gets to take him. He also was named a Smithsonian American Ingenuity Award. He, as all of you have heard, has given this TED Talk on his discovery that he'll be talking about today about creating false memories in the rat brain, or inception is kind of a more fun word to use. And so based on his publications in Nature and Science, what's the name of the academic recognition award that he has? That's what it's called. Okay, so some award for that. So his TED Talk, how many views do we have? Almost a million views for the TED Talk, so you all are lucky enough to hear the talk free of charge because of how it's called. Thank you, Steve, for coming.
Thanks everybody for attending. It really is my pleasure to be here and be able to share some of our stories on trying to find memories in the brain, turn memories on or off in the brain. It's the kind of stuff that for me still sounds very sci-fi and it kind of makes lab feel more like a hobby and less like a job. So I'm excited to actually share some of the nitty-gritty stuff that we didn't get a chance to talk about in the TED Talk because it really literally put everybody to sleep, so hopefully it doesn't do that. So we're going to get some jargony stuff out of the way really quickly. So the stuff that we study with memories was given a term a little bit under 100 years ago called engrams. So engrams is like, we call it the E word, the same way that people who study things like monkeys call consciousness the C word, because if you say it, it's a very loaded word. But I'm convinced that we're at an age now where we actually can begin to study things like engrams in the brain. So defining it really simply, an engram is just a memory. It's the total biological and electrophysiological realization of a memory, right? When you learn something, when you're forming a memory, whether it's studying for a class, memory of your first kiss, the works, there's stuff that happens in your brain. So the question is, how is that stuff biologically realized? The sum total of that realization is an engram. So we have a lot of engrams. One engram in particular that sticks with me, and it's really a testament to the kind of mental time travel that we do when you close your eyes and you just think back to literally any event in your life. I always make fun of my physicist friends for saying that we have the one thing in your brains that physicists would never have, which is a time machine to the past. And you do it effortlessly just by closing your eyes and thinking to the past. So a couple of years ago, I went to the Dominican Republic for spring break. Awesome idea. And I feel like a walking commercial for the DR. And I went water kayaking in the ocean, which was an absolutely terrible idea. Because when you're water kayaking in the ocean, it basically feels like you're on an aquatic treadmill the whole time, and you're not getting anywhere. So like any good friends, while I'm sitting here dying, all my friends were taking pictures of me falling over the kayak, not knowing what I was doing. So that's me. And when I'm thinking about something like drowning in the Atlantic, and actually the first thing that I had come to mind when this happened was that this is the real life version of the water maze that I haven't talked about. Because I had to put my head up and I'm like, where's the platform, which is the land, right, so that I can find it. It's one trial learning. So if you take a cross section of my brain and you ask, well, okay, where is this memory being processed? One of the most important areas, which I'm sure you've heard over and over again, is the hippocampus, right? Tucked just a little bit above your ears inwardly. So the question becomes is, how is the hippocampus doing this? How is it processing memories? Is it storing memories? What's its role? So one more memory that I wanted to share is, do you ever have to swipe in to get into like your dorms or to get into just anywhere? So in undergrad, we also had to swipe in to get to our dorms, and I have very fuzzy episodic memories. I'm trying to swipe in, I get to in the morning on Friday night, and this is exactly what I felt like, and this is equal level of PTSD as almost drowning. And these are hazier memories that we can get into afterwards. So again, how is the brain doing this? So, next credit, does anybody, oh, I wrote the name. Never mind, everybody knows who this is now. So, Wilder Penfield was one of the first guys to actually give us a really good idea as to where in the brain to look. Now we know that the hippocampus is important, but how do we know that, right? Like, how do we know that the entire jungle of the 80 billion neurons you have, where do you look for something as seemingly ephemeral as a memory? So, this was Penfield back, apparently he was an all-star quarterback in Princeton, which is not at all how I imagine neurosurgeons would actually look. So, what he would do is, you go into the brain of people with epilepsy, for example, and you go in and you scoop out the problematic parts of the brain. So what he would do is apply tiny bits of electricity to parts of the brain, and then ask the person, when you feel like a seizure is coming on, let me know, and I'll resect that part of the brain, right? You can be awake while this is happening, your brain doesn't have pain receptors. So in a subset of patients, when he would stimulate near the temporal lobes of this chunk of brain over here, and the hippocampus is embedded inwardly in it, a subset of patients would randomly start recalling memories or episodic autobiographical memories. They would say, I have no idea what's going on right now. For some reason, I'm remembering walking my cat, or my grandma baking me cookies on my 19th birthday, blah, blah, blah. And they would start recalling vividly these memories. So accidentally, Penfield discovered that parts of the medial temporal lobe, when you turn them on, are sufficient to reinstate that entire process of recollection. So that was in the 40s and 50s. So that gave us fantastic hints as to where the brain can begin to look if we want to try to play around with some memories. So the concept is really simple, right? You go into the brain, find a memory, turn it on or off. So that sounds fun. To do it, there's actually a lot of genetic trickery that we have to go through. So I'm going to go through this whole. Who's heard about the genetics? First of all, because was this discussed in class before? Yeah, great. So I'm going to go through optogenetics, which I'm convinced is revolutionizing neuroscience in the way that when Watson and Crick, for example, figured out the structure of DNA, that was a ginormous revolution that happened in molecular biology. So nowadays, optogenetics is something that was pretty much discovered around 2005 and is used in over thousands of labs now. And I mean, there's a lot of Nobel Prizes waiting to come out for it. And it's a fantastic tool because when you think about it, your brain communicates on the order of milliseconds, right? Most of the tools that we had previously to manipulate brain activity was it would take minutes or hours, for example, to actually have effects in the brain. So whether it's scooping out the brain drugs and so on. So nothing is faster than the speed of light, and optogenetics is basically a way of tricking brain cells to respond to pulses of light. So now finally we've upgraded. It's like you're upgrading the study of the brain from like the 56K internet days to like cable DSL BIOS now. You can actually communicate on the order, the temporal order that the brain communicates on. So this is what it looks like. You can actually trick brain cells to respond to pulses of light, and then you can shoot light into the brain and turn on a very specific set of brain cells, which when combined with the power of genetics, you can actually do some really powerful things. So the revolution that we're going through now in neuroscience is actually, it's really like a privileged pocket of time to be in because it's not every day science goes through a revolution like this, and we're all living through one right now with this. So it was a result of algae. So algae, right, they do this thing called phototaxis where they orient themselves towards or away from light. But that's really neat. How do they do that? That means that they're actually transducing light signals into some kind of movement. So this is one algae, this is the other one, and we'll talk about both because both are the two things that were accidentally discovered in nature that now change the way of how we try to perturb neural activity. So one of them here, it's abbreviated, so one of them is called channel rhodopsin. We spell that in a subsequent slide. So Channel rhodopsin, it's called channel because it's a channel, it's a pore that embeds itself in the cell membrane. Rhodopsin, because like the rhodopsin that you have in your eyes, it's responsive to light. So channel rhodopsin is a kind of pore that normally is closed. If you shoot blue light specifically at it, it opens up, it lets in sodium, action potentials happen. So really straightforward. All it is it's just think of the sodium channels that we learned about when we were learning about action potentials. Now this is just a light-sensitive channel. So you shoot light, it turns on. You take the light off, it closes. So it's millisecond precision. The other one that I won't talk about too much, but it's just as an FYI, it's called halo rhodopsin, MPHR for short. So MPHR is because of the bacteria that it's derived from. So halo rhodopsin is just the opposite. Rather than letting in sodium, it's actually a pump that pumps in chloride into the brain cell. So chloride, if you remember, has a negative charge to it. So if you pump in negative charge to a cell, you turn it off, right? You hyperpolarize it. So that's kind of amazing because that means that now you literally have an on-off switch for brain cells. So here's just action potentials, for example, in one brain cell going at 5 times per second, 10, 20, 30 times per second. That's a result of just shooting light, channel rhodopsin opens, the cell begins firing at whatever frequency that you're shooting the light at. Similarly with halo rhodopsin, you just give yellow light now. I should have mentioned that before, these respond to different kinds of light. You give yellow light and you can shut off the brain. So this is sort of the ace in the sleeve that we needed, that if we wanted to do something like turn on a memory or turn it off, when you close your eyes and you think of a memory, it happens really fast. So we can go in now and trick brain cells that we think are holding on to a particular memory. We can trick those brain cells to express something like channel rhodopsin or channel rhodopsin itself, shoot light into the brain, those brain cells turn on, does the memory come back or not? So this is what I mentioned before. Channel rhodopsin again responds to blue light, halo rhodopsin responds to yellow light. So this is just what it looks like. This is actually one of my mice that shooting blue light into the brain or green light into the brain. That's a whole other project that I can get into if we have time. But any questions for now before moving into equally complicated stuff? No. So okay, again, the question becomes, can we go into an area like the hippocampus, which thanks to Penfield and a bazillion other experiments we know is important for memory, can we go into the hippocampus and find not just every single brain cell, right? Because if you indiscriminately turn on every single brain cell, what happens is you're going to get a seizure, right? So this is why the whole 10% myth thing is myth, because if you, it's like, have you guys seen the movie Limitless, for example? It's actually a fantastic movie, but one of the things that drives me bananas and every other neuroscientist is like, what if you have this pill that can activate 95% of your brain and you can be Scarlett Johansson in that other terrible movie? And what it ends up happening if you have a drug that does that is you literally have the most massive seizure known to mankind. So that's not a good thing. So rather than giving our mice the most massive seizure known to mankind, you could go in and find cells that we think are specifically holding on to one memory. So this is probably the most important slide in the talk. It's going to go from 0 to crazy complicated really fast. So stop me at any point if it's unclear. But I do think that this is a powerful system where we can actually open and close windows, for example, of when an animal forms a memory and finding the brain cells in that window that are active when the animal is making that memory. So I'll become clear in a second. So the ace up our sleeve, like I said, was channel rhodopsin, right? We can trick brain cells to respond to pulses of light. So all we do is we can genetically insert into the genome the gene that encodes for channel rhodopsin. These cells now express channel rhodopsin on their surface. We shoot light into the brain, channel rhodopsin turns that brain cell on. So that's the part we just reviewed. Now, like I said, if you don't want to be Scarlett Johansson having massive seizures, you have to find the brain cells that are holding on to just one memory. So how do you do that? It turns out that whenever a brain cell fires, so as soon as it starts turning on, there's 1,001 things that happen. One of the things that happens is that there's information in the form of chemicals, like sodium, chloride, and so on, that actually reach the genome. So there are a specific set of genes that are turned on only when a brain cell is turned on. So we call them activity-dependent genes, just because their activity depends on the activity of the brain cell. So if a brain cell turns on, corresponding set of genes turned on. If the brain cell is off, those genes are not turned on. One of the genes that I'll talk about a lot is called CFOS. So the name itself isn't important, it's just that this is one of the genes that actually knows, quote unquote, when a brain cell is turned on. Because the brain cell fires, and then CFOS now begins to, the gene gets turned on, it undergoes transcription, translation, it works. So here's what a cross-section of a mouse hippocampus looks like. And the green dots are tiny brain cells that are expressing CFOS. So we can go in and actually visualize the brain cells that were active. So for example, if we put the animal in a box, give it a couple of shocks, it's forming a memory of that box. That means a corresponding set of cells in the hippocampus are going to be active, right? So that means we can go in and ask which brain cells are active. How do we know? We look at which ones are expressing CFOS. Those are the ones that were recently active. So, one thing though is, how do you know, like an animal undergoes a lot in its lifetime, right? Like how do you know that the brain cells that are turning on are in response to one specific event? So, it turns out that the key part of CFOS is actually a very real biological sensor, a sensor that senses when a brain cell turns on. It's actually the promoter, the initial segment of the gene. That promoter of CFOS is the biological sensor. That's what knows when the brain cell is turned on. So with a little bit of genetic engineering, you can imagine, what if you just cut and paste? You cut the promoter of CFOS, and then you paste it onto the gene that makes channel rhodopsin. That means whenever a brain cell is turned on, the CFOS promoter will be turned on, and then that'll drive the expression of whatever is attached to it, such as channel rhodopsin. The problem with that is that if you start moving the mouse like in a cage, you start juggling it, you put it in anything that's not your memory task, brain cells are going to become labeled, right? There's no way of opening and closing that window, a defined window, so that you can say, everything that happens to the mouse up until this point is not going to get labeled with channel rhodopsin. Everything hereafter will be labeled with channel rhodopsin. So to do that, and this is the most complicated part, and again, the names aren't important, it's what they do that's important here. So what you can do for that, to actually open and close windows of when you can label brain cells in the hippocampus, you can cut the promoter of CFOS, so you do some genetic trickery, you cut the promoter, and then you paste it to what's known as TTA. So it stands for tetracycline transactivator. The important part is the transactivator part, because it gives away what it does. So in biology, usually when something is trans, it means it travels somewhere, right? So transactivator, it travels somewhere and activates something. So let's say a brain cell is active, it turns on the CFOS promoter, it drives the expression of TTA. So TTA now is in its protein form, trans, it's traveling somewhere, it's about to activate something. What does it activate? It activates whatever responds to it, right? Which is conveniently named the TTA response element. So it's just another sequence of gene. A brain cell is active, TTA turns on, TTA travels and activates TRE. So the important part here is that when TRE now is turned on, this just acts as another promoter. It just drives whatever's attached to it. What can be attached to it is literally anything. And in our case, we happen to make it channel rhodopsin. So there's a whole cascade of events that happen. And the question is, if this is confusing, by the way, this took me a year of grad school to even understand what these terms mean, so don't worry. The important part is that this is the other ace in the hole that we needed, because this process of TTA binding to TRE is controlled by a drug called dox, or doxycycline, which we might have heard of before. It's an antibiotic. So when doxycycline, you can give it in the animal's diet. So you give the animal doxycycline, it just eats it all the time. This process stops, because dox binds to TTA and inhibits it. So you can imagine that if we just say, the jargony way of saying it is that an animal is on dox. So if you raise an animal on dox, a cell can become active, TTA is made, but because it's on dox, it binds to TTA and prevents this step from happening. So nothing is labeled in the brain. So let's say tomorrow we want to go and do something like fear condition the animal, to put it in a box and give it a couple of shocks. What we can do is we say we take the animal off dox. We remove dox from the diet. That opens up the window now for a cell to become active, such as when the animal is being fear conditioned. That activity now drives TTA, it binds to TRE, and drives the expression of ultimately what will be channel rhodopsin. And then you can put the animal back on dox, and no more labeling happens. It's a permanent label that stays there at a defined period of time. Raise your hand if you're completely confused. Okay, raise your hand if you understand it. Right, great. So that's the system. If you have to read any of the papers, it's a very commonly used system in genetics now, and in animal behavior, because again, you can open and close windows of when to label brain cells with any protein that you want. Ours happens to be channel rhodopsin. So okay, for the long, I'm a fan of naming things. So for the longest time, during lab meetings, I would argue with my supervisor, can you please name this something cool, because I think it's a neat system. So, what I really wanted to name this was rapid engram labeling using a doxycycline suppressible and not the genetically modulated C+-driven knocking system, or Red Sox for short. Everybody, I was born and raised in Boston. I'm like contractually obligated to be a Boston sports fan. If you're from New York, I'm sorry, I'm not sorry. And this didn't fly with our supervisor, which makes no sense because we had the lab had a paper like in 2004 where they named the technique Dice K, which is after a Red Sox pitcher. And like if you can name it after a Red Sox pitcher, why not just call it Red Sox? Anyways, now speaking of contractually obligated, every single talk I give, I have this slide hoping that it'll catch on. It hasn't really caught on in like 4 years, but I'm still trying. So when all is said and done, what does it look like? So let's say we take our animals off docks, we open that window, we put them in a box and give them a couple of shocks, mild shocks, so that they form memory of that box. Then that whole process that I just showed you happens, ultimately leading to the expression of channel rhodopsin, only in the set of cells that were active when the animal was learning something. So what you're looking at here is a cross-section of the brain, and the green dots are the ones that are, in this case, channel rhodopsin positive. The channel rhodopsin is actually, it's also tethered to GFP just to make it fluorescent green. So all the green cells are the ones that are active when the animal was learning the fear memory. So I'd like to think of this as a nice romanticized, not really that romanticized way, of whatever a memory looks like at the level of a brain, you're looking at a tiny cross-section of it. Like these are the brain cells that are holding on to one memory, a fear memory in the brain. Some aspect of what it looks like is this. So okay, the first project, we have to put this to the test now, right, and do this $1 million experiment. Can we put the animal in a blue box, we'll just call it environment A. It's off docks. We give it a couple of foot shots that it forms a memory. It's a specific set of cells now in the hippocampus that have channel rhodopsin, right, because of the whole system that we talked about before. Those are the brain cells that are now responsive to light. So normally, when you put the animal back in environment A, where it was given a couple of shocks, when you put it back there, freezes, right? It shows fear behavior, it goes to the corner, it huddles, it's completely immobile. So if you put it in a completely new box, the animals go, they sniff around, they mind their own business, they're moving around the entire time. So the $1 million experiment becomes, what if when they're in this box that they have no reason to be scared of, we shoot light into the brain? Does the animal think that it's back in the environment where it was shocked? Because the prediction is, if it does think that it was in the environment where it was shocked, it should freeze, right? It should show fear behavior. This was the very first animal that we ran. It was a... So this was the very first animal that we ran. It was a cold March 23rd. So, there's nothing. That awesome. So the animal, this one is on docks, meaning nothing is labeled in the brain, right? Because a very valid criticism is, what if you just shoot light into the brain and that freaks the animal out, right? And that's why it's freezing, is because it has lasers going into its head. As you can see, when you turn the light on, the animals really don't care. They just keep sniffing around, they keep walking around. Now, this one has a fear memory labeled with channel rhodopsin in the hippocampus. And it's in an environment where nothing bad happened to it. And as soon as you turn on the light, you see the animal go into that immobile fear behavior, that freezing posture. I had one of the biggest hangovers of my life the next day after that worked, because it was like 2 years worth of this one animal, is this going to work or not? And then of course we go and do it with other animals and we produce it into 1 billion animal controls and argue with reviewers and go through that whole process. I felt like this. I was just completely... honestly, like, it was one of those responses that when the experiment was done, I was just like, holy shit, it actually worked. Like, you can tell, like, it's very obvious. Like, that's why we use freezing behavior, because the animal is either moving or not. It's very binary, so you have your answer very clearly. So what the data actually looks like is this. So here's this cross-section of part of the hippocampus that we think is holding on to this fear memory. Again, the green is channel rhodopsin. So what we can do is, the blue is the animals that did not have the fear memory label. It was just shooting light into no channel rhodopsin, no nothing, just brain tissue. That's a very important control variable. Now, the red ones are the ones that do have the fear memory labeled with channel rhodopsin. You can see that during off, we're measuring freezing on the y-axis. When the light is off, they're not freezing. When the light is on, as you saw in the video, they freeze pretty robustly. You turn the light off, and it goes back to baseline. Turn the light on, it starts freezing again. So it's a very real switch on turning on fear memory. Now, in my opinion, the single most important control, because this right now, we can exclude that it's because light is having some indiscriminate effects, right? Because we have this blue group as a control. But if you're an astute reviewer for this paper, you can say, well, technically, you haven't shown that it's a fear memory per se. Because what if just by activating the hippocampus, let's forget the whole fear memory part. What if just by activating the hippocampus, you're freaking the animal out? Like it feels something fuzzy happening in its brain, and it just starts freezing because it doesn't know what's going on. That's a very valid concern. So the way that you can control for that is, rather than labeling a fear memory, you just label a neutral memory. You put the animal in a box, and it just sniffs around, it learns about the box, it forms a memory about the box. Conveniently, the same number of cells are labeled in the hippocampus with channel rhodopsin in response to that kind of memory. And we do the same exact procedure. We just shoot light into the brain. And this was, in all honesty, this is actually when we really celebrated, because this is when we really knew that if you activate just a random population, a random set of hippocampal cells, nothing happens to behavior. But if you activate the set of cells that we think are holding on to a fear memory, then you can recapitulate the freezing behavior and recall that memory.
How did you get to like that location being the fear memory?
So a lot of it, there's a couple of things. One was from previous work just suggested that the hippocampus was really important for memories in general. And then this particular area that we were focusing on in the hippocampus is remarkably sensitive to changes in context. So if we're looking for the memory of an environment, for example, it made sense to start at the area in the brain that's very sensitive to changes in the environment. It's very much involved in processing and in the environment as a whole, and is sensitive to subtle changes. So the conclusion from that, from the first study here, is that indeed, like, you can actually go into the brain. You can find a set of cells that are holding on to one memory, trick them to respond to pulses of light, and flick on that memory. So the next thing is, I love Christopher Nolan movies like crazy, so we call this one Project Inception, because I really was like enamored with this idea of, well, if we can turn memories on, can we actually modify their contents? So one important thing that I'm sure everybody knows is that memory, or I hope everybody knows, memory is the least reliable thing like in the world. Like every memory is only as real as the last time that you recalled it. Like every time you recall a memory, for the most part, you're constantly modifying it and updating it with new information. And it's a seemingly like, you're not even consciously aware of updating the contents of memories. But every time you recall it, you know, grandma's sweater goes from purple to blue to yellow. You get into arguments with your significant other about where you left the keys. One person remembers it one way, the other one remembers it the other way, and so on. So taken to its extreme, things like false memories can form, and memories can become warped over time. So one thing I thought was, okay, if we can flick on a memory, then can we flick it on and update it with new information? In essence, trying to create a kind of false memory. So if everybody could just stare at the cursor. If you've seen this kind of task already, don't say anything so it doesn't ruin it for everybody. So I'm going to flick through a bunch of slides really carefully and just pay careful attention. Everybody get it? I'm describing what's going to happen in like 20 minutes. So, just very simply, raise your hand if you saw this word. Raise your hand if you saw this word. Raise your hand if you saw this word. This word? This word? This word? Right, and that word. So, this actually worked out really well. So, for the most part, there's a fraction of people, maybe about a third usually if it's done particularly well, These are the three words, of course, that you didn't see on the list. Locke and Godwin. Beer was not on the list, but if there's a theme there, right? It's very easy to think that it was on there because there's a commonality in that set of words. So I'm projecting like way too much of my personality in this slide. But this is something that it's a very mild effect, but it's a very benign kind of false memory. But it still gives us a window into the kind of stuff that's happening in the brain when you're forming a false memory. It turns out when you're recalling these kinds of false memories, you actually see a lot of hippocampus activity. So that's kind of strange, because the same thing that lets you recall memories is also the same structure that enables the formation of false memories. It's a kind of evolutionary hiccup that happened in our brains, but it's a very profound one. So, project conception in a nutshell was to update the contents of a memory. So what we do is, let's say we take the animals off docks, right? We open that window for labeling, we trick brain cells to respond to pulses of light. We'll put them in this blue box, and we label a subset of hippocampus cells with channel rhodopsin, right? So now we have optical leverage over the memory of the blue box. So the most important group here is just the blue group. You can ignore these controls, we can talk about it afterwards. So if you look at freezing levels, when it's just exploring the blue box, they should be low, right? It has no reason to be scared of it. Now we put the animals back on docks, so nothing becomes labeled in the brain. You have the label for the channel rhodopsin for the memory for the blue box, but now nothing subsequently can be labeled because the animal's back on docks, right? TTA can't bind to TRE, jumpstart that whole process. So we put the animal in a green box because now it has two memories. It has a memory of the blue box and the memory of the green box, but only the blue box is the one that we can turn on with light, right? That's the prediction. Freezing levels, of course, are low in that group as well. It has no reason to be scared of the green box. So the quote-unquote false memory protocol is, now you put an animal, once again, the same animal, in a very different box. So a red box. It's never been in here before. And you fear condition it in the red box. You give it a couple of foot shocks. Now, the $1,000,000 intervention here is when you put the animal in the red box and you give it a couple of foot shocks, while it's getting those foot shocks, you shoot light into the brain so that, according to that first paper, it's recalling the memory of the blue box, even though it's in the red box. So it's in the red box, it's being fear conditioned, but we're shooting light into the brain so that it thinks it's in the blue box, and we give it a couple of foot shocks. So the prediction there is, if the animal is truly thinking about the blue box, then it should show fear behavior to the blue box, right? So the way to rigorously test that is, first, we put it back in the green box. Because anybody could say, well, you just fear conditioned the animal and shot light into the brain and confused it, it's going to be scared of everything that you do afterwards, independent of that memory. But when we put it in the green box, again, freezing levels are very low. It's not scared of the green box. And then the hangover 2 that happened was a result here to when we put the animal in the blue box, because this was the actual big test. This actually did happen on December 24th. I tried explaining this to my family at Christmas dinner. My brother and sister are lawyers. We speak completely different languages, so I think they were just rolling their eyes the entire time, like, oh my god, like Steve's talking again about stuff that nobody cares about. So I was kind of humbled during Christmas dinner. But later, it would become exciting. So I'm not bitter, whatever. So when we put the animal now back in the blue box, the prediction is that it was thinking of the blue box while being fear conditioned in the red box, right? And then the blue group shows a lot of freezing only to the blue box compared to controls and so on. So I think this video should work. Also, this was the first animal that I ran before Christmas dinner. Okay, so it's in environment A. It's forming a memory of this box right now. The memory of that box is becoming labeled with channel rhodopsin in the hippocampus. This is the blue box, quote unquote. So now it's being fear conditioned in the red box, but we're light reactivating supposedly the memory of the blue box, the one that we just saw, right? So the animal gets a handful of mild foot shocks. It's like static shock is what we heard them. And then when you put it back in the blue box, that's what they look like. I think it's kind of cool. Okay, how are we doing on time? I completely lost. Yeah? So the very last thing, and this is a project that I'm working on right now, and I'm not going to go over too much of the data for the sake of time. If you noticed, while I was over there, while Evan was talking, I was kind of like angry looking at my computer. We literally just got reviews back for this paper that we submitted a few months ago, as I was sitting there. So like I need to go and like really digest all of it. It was good news because all we have to do is arguing and not many experiments. So that's good. The project that I wanted to focus on next was, it's memory manipulation is cool, kind of scary. We can talk about all the ethical ramifications afterwards. But I thought, why not finally begin to re-aim this technology to the realm of psychiatric disorders? Can we use memory as sort of a weapon to try to combat certain psychiatric disorders, such as depression, anxiety, mood disorders, and so on? Because we got a flurry of like emails and people and like journalists asking, when am I going to be able to do this to suppress the traumatic components of a PTSD a PTSD episode, for example? Like when can we go in and actually suppress the traumatic components of that memory? Or if you've had traumatic breakups like I've had, like when can you go and just erase that like eternal sunshine? So like, so one of the things that we thought was, can we actually go and try to intervene with psychiatric related states in animals and use this memory manipulation technology? So the 1950s was an absolutely wonderful decade because the entire field of psychopharmaceuticals also underwent a revolution. The way optogenetics is going through a revolution around neuroscience, the way the structure of DNA made a revolution in molecular biology, or the Beatles in the 60s and so on. Psychopharmaceuticals went through a revolution in the 1950s. We finally found targets that can actually go into the brain and maybe suppress certain symptoms of depression, anxiety, and so on. And then the former head of the NIH said that revolution suddenly stalled. So the question is, what happened? Since the 1950s and 60s, most, but not all, most drugs were what he calls copycat drugs, right? SSRIs and things like Prozac, everything was just an iteration of what we discovered accidentally in the 1950s and 60s. So what happened is that people stopped thinking outside of the box, and then luckily, and as you heard from Evan's talk, now we have new drug targets now. There's better drugs, and they're not just copycats of the 1950s drugs. They're new things. So to complement that, I thought, why not go into the brain and harness the brain activity itself? Try to see if we can use memory as a weapon, like I said. The big hypothesis here, for example, was we have like another paper that recently came out. It was a postdoc and a grad student with this dynamic duo in the lab as well, who, among many things, showed that instead of incepting false fear memories or reactivating fear memories, you can go into the brain and the hippocampus again and reactivate positive memories. So it's just nicer for the mice. It's a positive memory. So there's a lot of positive memories for mice, right? You can give them things like amphetamines or cocaine or so on and so forth. But the one that actually gave us the most robust, whatever it is to have a positive index from the animal, was actually exposing male mice to female mice. Male mice go bananas when you expose them to female mice. So when I was presenting this at lab meeting, every guy was just like, oh, well, yeah, obviously that makes sense. Like, so when you all you have to do is you take the animal off docks, right, to open that window, put a couple of female mice in there, they go bonkers, and then you put the animal back on docks to prevent any further labeling, and what you can do is something like shoot light in one half of the box. Because if you put a female in that half of the box, the animal spends a lot of time in that half of the box, right? It shows a preference. So we just call it a place preference assay. So rather than that, we shoot light only when the animal is in this half of the box, and the animal shows a strong preference for that. And if it's a fear memory, it shows a strong aversion to it. So that was neat because it means that now we actually have a new tool to go into the brain. And what if we can use positive memories specifically to try to suppress some symptoms associated with depression? So that, in this case, is the $1 million hypothesis, is that positive memories artificially activated are sufficient to mitigate or suppress some of the symptoms associated with depression. So the huge disclaimer here, mice are not humans, right? So there's a lot of things, and this is really important, there's a lot of things that we can and potentially can't model in animals. So things like guilt, suicidality, sadness, we don't have animal models for that. We don't even know if animals can feel that. And if they do, we don't have a good behavioral readout of what that looks like. However, there's other things like motivation to approach naturally pleasurable stimuli, such as sugar water or another female. That's a pretty good readout of the animal's hedonic state, right? Anhedonia is the lack of that. So just motivation to escape from a challenging situation, right? Does it want to get out of bed and have Ben and Jerry's or not? So that's something that we can begin to at least somewhat model those symptoms in animals and without getting into the nitty gritty. This is just what it looks like. This one here is sugar water, the one behind it there is regular water. Here's the animal. It's plugged into the optic fiber, which will eventually shoot light into the brain to reactivate the positive memory. The short of it is, yeah, it does seem to actually suppress a handful of symptoms associated with depression in as far as we can model those in animals, whereas other symptoms remain untouched, which is interesting because it's a specific kind of intervention. I can go over all of this afterwards, but the point of it is that now we can actually begin to use this kind of technology for the sake of both basic science and in this arena of psychiatric disease disorders that we can model in animals. So that's basically it for the data part of it. Evan gave an awesome overview of therapeutic value, for example, of developing certain drugs to try to combat certain things like Alzheimer's. I want to give now sort of a big rally for just basic science itself as well. One of the most common questions that I've faced in grad school, for example, is like, when is this going to reach people? Again, like, when can you suppress the parts of the traumatic components of PTSD? When can you suppress some symptoms associated with anxiety or schizophrenia or OCD or so on? So that's an important question, and we need that work happening in parallel to basic science. But I think that if that's the only thing that the person asking that question is concerned with, it's a little bit short-sighted, because Mark Twain has this great quote that history doesn't repeat itself, but it does rhyme. So we have a lot of rhyming that's happened, especially in the 1900s in science, that we can learn from. So this is basically to convince everybody here to do basic science. So there's examples of where basic science has changed everybody's life, and you probably don't even know it. So one example, for example, is, okay, so you're in the 1970s and you're a group of physicists and you're pointing your telescopes up to the stars and you want to figure out what's the composition of these stars, how do these atoms move the way they move, blah, blah, blah. A lot of math happens, you publish a paper, one and done. Now, a decade later, brain enthusiasts came in and said, actually, these algorithms are really useful because rather than pointing that telescope out there, what if you pointed inwardly and bam, like the fMRI is invented based off of that? Did those physicists care that their technology was going to enable the fMRI? No, they couldn't even predict that. They just cared about the composition of stars, they figured it out. Basic science then turned into application in the course of a decade. Even longer than that, I think it was in the late 90s where the Hubble telescope had this big oops where one of the mirrors was kind of curved and foggy. And they had to develop algorithms to fix that mirror because the Hubble telescope was really expensive. So computer scientists sat down, they developed algorithms that basically fixed the images that the Hubble was bringing back, and they made the resolution on them better. Those same algorithms are used now in hospitals, again, pointing it inward. They give you better resolution images for breast cancer screens. The people who developed these algorithms for the Hubble had no idea that that was going to be used today for breast cancer screens to give the doctors better resolution images. And yet that happened. So asking those people, why are you fixing the Hubble when you have problems on Earth, is a misguided question because it's just a longevity game. In time, that'll become applicable. My favorite example, and the last one, is, so okay, it's the early 1900s and you're Einstein, and all you care about is nerding out and figuring out how the curvature of space-time happens. Why does it happen? So you go, you figure it out, you completely revolutionize the field because you figure out stuff about how, general relativity and gravity and curving space and yada, yada, yada. So he figures it out because that's what he cared about. He cared about just the nature of things. So today, those exact same equations that he used to figure out the curvature of space and time are, well, here's used Google Maps, for example, in the past month, just to get from point A to point B, right? We use it to get here today. Those exact algorithms are used to help send your signal from here to a satellite and back and take the curvature of space-time into account. Because if we didn't have Einstein's relativity, the signal that would come back would be off by 50 kilometers and grow exponentially every single day. So I always say that when Apple Maps first came out, they didn't take those algorithms into consideration because Apple Maps was awful when they first came out. But that's the point here is that, did Einstein care about the iPhone that's in everybody's pocket right now? No, right? He wanted to figure out just stuff about space, and he did. Granted, it took 100 years, but now it's probably one of the most widely used things that people have in their phones, and it's very useful too. It's not just flashy coolness, it's actually something that helps you get from point A to point B and makes your life infinitely easier. The the moral of the story is that basic science will matter. It's just a matter of when and how it matters. And maybe asking when and how is a myopic, short-sighted question. It's just a matter of knowledge is power in this case. So why not discover for the sake of that knowledge, because somebody else will come along and use it for breast cancer screens, or use it for the GPS of your phone. Okay, so I've asked myself this question so many times when I was transitioning out of college and I was trying to figure out what in the world am I going to do with my life. So the process of going through grad school and entering grad school is pretty fresh in my mind still. So if you have any questions, whether it's about the process of applying to grad school, how do I know that I want to do grad school, why not med school, blah, 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 feel free to send me a billion and one emails because I'm happy to help out with it. Again, because it's still pretty fresh in my mind, and then I can tell you about my lab experience in undergrad. The first one was awful, the second one was awesome, and then just how these things even became, like how I even became interested in neuroscience. And the short answer is that a lot of it is, J.K. Rowling has this great quote, and I've never read any of the Harry Potter, so whatever, but she has this great quote saying that I don't believe in fate, but she believes in hard work and good luck, and that a lot of the former leads to a lot of the latter. That's exactly what happens here, is that you work hard, and then the doors open, and you embrace that kind of luck. So I'm happy to share those kinds of stories, because it wasn't just, it's not something that, I mean, you both work hard for it and take advantage of those luck opportunities that come about throughout college and thereafter. Anyway, so I probably went over, so that's it. Thank you. Do you have time for questions? Or
Yeah, a little bit of time for questions. And then if anybody has follow-up questions for us as well.
No one's worried about my manipulation. Or the questions also don't have to be specific to why CFOS, CTA, blah, 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 but it could also be more about, again, the transition either out of college or in grad school, what's grad school like, all that fun stuff. Thereafter, the gazillion and one careers that a PhD actually enables, contrary to what every other PhD will tell you. And sorry if I'm like blaspheming right now, but yeah, any questions, whether it's career-oriented, science-oriented, do I have to worry about inception happening to people or so on? I guess the answer is yes. Okay. Yep.
So what would you say is the next step in your research here?
Yeah, I applied for jobs is number one. Now the big thing is the depression stuff is actually something that I really want to work on, because we have one finding, for example, where a big criticism of optogenetics is you turn the light on and the animal shows some kind of behavior, whether it's in response to a positive memory or freezing. But you turn the light off and it goes away, right? So what's the therapeutic value of that? Like, you're switching it on. You can't keep light on in the brain forever because you're just going to fry the brain. So one finding that we do have in this paper is what we did was we tried to mimic the course of drugs. Many of you know that if you give a person Prozac once, it doesn't really have an effect. You have to give it to them chronically over weeks for it to actually precipitate effects, right? So what we did was we went into the brain and reactivated this positive memory multiple times a day for multiple days. And then that actually has long-term effects, so that, like, let's say we reactivate it twice a day for 5 days. On days 6, 7, and 8, when we're doing all of our behavior tests and everything, there's no light in that situation, but the animals actually show a rescue. So they no longer show some of these depression-like behaviors. So something that I wanted to focus on next was trying to figure out why that works. How does that happen? Can we use optogenetics not just to turn stuff on or off in real time, but can we use it to actually induce plasticity in the brain over long periods of time? Because then you have an intervention now that you do for, let's say, 2 weeks, and then subsequently your animals show a rescue. That, I think, translates better than having to figure stuff out in real time. So one part is to try to do this kind of chronic stimulation protocols, and then figure out what does that tell us about the brain? Why would the animals rescue? And then the other ones, I do want to go into a little bit with the stress and PTSD angle as well, because there are different corners of the brain. It's been proposed process memories, different aspects of memories. So I want to actually go in and look at animal models of things like PTSD, for example, and like I said, try to shut off the actual emotional oomph that PTSD memory has, but still keep the contents of the memory intact, just kind of begin to really suppress the emotions involved in it. So I think those two things hopefully will get me tenure in 7 years, and I'll give you an answer.
That's great.
Yep.
What's the stuff that you put in your food again?
I'm sorry?
What was the stuff that you put in your food?
Oh, dogs.
Yeah, so when you give the animal dogs, are the memories just going to like a different part of their brain, or do they not have the memories?
So the dox itself is the dox itself is actually very lipidy because we need the lipidy to the lipid stuff to deliver it into the brain and into the body. So the only thing that happens with dox is that it's just an other part of the animal's diet that's included there. For the most part, we don't think that there's any biologically specific effects that the animals undergo because they have dox in their diet. And we give dox to all the controlled animals in that too. So for the most part, all it's doing, as far as we know, is just actually controlling that window of when we can actually begin to label memories or not. But there are, it's a really good question because that is something that one of our reviewers asked, for example, is just how do you know that dox isn't just doing something else to the animal that you're not sure of because it is in the animal's diet. And the only way to really rule that out is to also give your animals the exact same diet as well, or the control animals the exact same diet, because then it's a fair comparison across the groups. That's on the other hand.
Other questions also for Evan?
Yeah.
Okay, I think we'll call it then. Thank you again so